Okay, so thanks to ProImmune for inviting me. I'm kind of an outlier in this whole conference. I'm not an immunologist, I'm a geneticist, and I'm going to be talking from the engineering side, not the detection side. Okay, so here's what I'm going to talk about today. I'll spend a couple of minutes telling you about our company, and then the platform that we are designed around. I'll do a little proof of concept on that platform. We'll spend one slide on intellectual property, okay, and then how this would apply to deimmunization. Okay, so this is just a quick overview of our company. Essentially, we're a protein engineering company, so not an immunologic company. Okay, and what we've developed is a high throughput, what we call mapping platform, and what it's used for is quantifying the properties of hundreds or thousands of molecular variants at once. Okay, provides a comprehensive map for engineering proteins and peptides, and it's got multiple applications for generating Biobetter Therapeutics. Okay, so the setup of the system. So I'm not sure how many people are molecular biologists today. This is going to be mostly molecular biology up front. But basically what we usually do in molecular biology to engineer proteins is we create large libraries. We select out and characterize individual variants from those huge libraries. Okay, and what we're going to do here is we're going to try and quantify the effect of every single possible amino acid change you can make in a protein binding site. We're going to try and do them all simultaneously. Okay, and what we're going to do is generate a map of the binding surface, and that's going to allow us to engineer a wide range of different features into that binding surface. Okay, so here's basically the concept pictorially, is you've got a large protein binding site, okay, and instead of making a huge library to look at that protein binding site and lots of combinations, we're going to make single amino acid changes. Okay, and we're going to quantify every single one that we can make. So the one way to look at the data is a heat map like this, and we'll read the position of the amino acid change here, which represents all these amino acids that are colored in blue, and we'll look at every single possible mutation you could make using the code, so all 20 different amino acid variants. Okay, we'll characterize them all in one fell swoop, and what that's going to let us do is read the power of each of those mutations with respect to binding. So obviously we can look at affinity, we can look at cross-reactivity, but if we've mapped immunogenic peptides, okay, we can deimmunize them, we can humanize much more efficiently, we can look at stability, and we can look at half-life. Okay, and here's kind of the key right here. Instead of creating a huge library that represents this binding site, we're going to create a rather small one. Okay, and so if we look at an antibody binding site, we've got 50 to 80 amino acids in the CDRs of that binding site. Okay, we've got 20 possible amino acids at each site. And so we're looking at libraries in the small numbers of thousands, not in the large numbers of billions like antibody libraries usually look like. Okay, this slide is just a kind of recent view. Basically, this is the way genetics has always been done, but by looking at amino acid mutations one by one. Okay. More recently, there's been a few companies that have looked through amino acid mutations, multiple changes at a time. Okay, and so single point mutants. Okay, BioRen was one of the first. They used a yeast display system. They created large numbers of, well, thousands of mutations at once. Were able to screen them rather quickly using the FACS. Okay, but then they had to analyze the sequences one by one. Okay, DeRivo did something similar with a different detection system and basically Fab secretion. Okay, and what they did was, again, screen them rather rapidly for binding, but then they were forced out the backside to analyze the sequences one at a time. So you were really just using a high throughput screening method, but still were slowed down by your sequencing. At PDL, we set up a system in my group, and Fiona was part of that group, that essentially, the goal was to deimmunize Humira and a few other molecules that we had in mind. Okay, and the big difference was we created these single amino acid change libraries, but analyzed them in mass using next generation sequencing. Okay, recently you've seen a publication from that. You'll see a publication from PDL, which is now Abbott. In the near future, that'll be in Journal of Immunology. But there was a very nice paper from University of Washington, David Baker's lab, And they looked at a similar sort of system with the yeast display, and that contrasts with our system that we have now at full spectrum that is essentially phage display, which offers a number of kind of unique advantages. Okay, phage, very hardy. Okay, you can apply them to many different assays. They're resistant to pH, they're resistant to temperature. They're essentially not alive, and so we can incubate them for long periods of time and look at equilibrium binding assays. And up at top, most of the molecular biologists will recognize this is typical phage screening. Okay, we create huge libraries in the billions or above. Okay, we bind them to the surface of proteins, usually in ELISA wells. Okay, we do stringent washing through multiple rounds, but then we're stuck at the end analyzing clones one by one. Okay, and so you start with huge libraries, but you wind up with small quantities of data. Okay, and you've gone through multiple rounds of selection, and so, you know, the classic in genetics is you get what you select for. If you really know what you're selecting for, that's great, okay, but in these systems, it's not just binding. Okay, we've adapted this system to our purposes, okay, and so we make much smaller libraries, okay, on the order of thousands. Okay, we do kind of standard equilibrium binding reactions, and now we can rescue both sides of the binding reaction and analyze both to completion. Okay, we can analyze the bound molecules, we can analyze the free molecules, and we go to a next generation sequencer. We can read the frequency of each of those molecules at fairly high efficiency. We're getting millions of sequences out of this sequencer. Okay, that allows us to generate a heat map that basically tells us what the frequency or the binding efficiency of every mutation in that library was all at once. Okay. So what we did to test this system, this was just the design. What we did to test is go after a molecule that was already very well characterized from the molecular standpoint, okay, and that was Avastin. Okay, what we know about Avastin is it's CDR3 of the heavy chain is the major binding epitope of that molecule. Okay, we created a library that included that epitope and basically some way down both sides of the framework. Okay, we expressed the library as bacterial Fabs. One thing I should mention, that's a 25-position library, and we included all 20 amino acids at each and every site, one by one in each phage. Okay, we expressed the library as Fabs. We ran equilibrium binding reactions against its target, which is VEGF. We sequenced the bound in the initial phage populations. We can also sequence the bound in the free populations to normalize even a little bit better. And that allowed us to statistically calculate the relative binding efficiency of every variant in the library. Okay, one way that we can characterize this data fairly quickly is we create these libraries through NNN mutagenesis. Okay, and so we create lots of silent wild type variants in the library. Okay, the nice thing about silent wild types is synonymous codons is essentially they all target or encode for the same protein, and the same protein should consistently behave the same way if we're going to expect to use this system. And that's what we see. Okay, and so what we've got is our binding ratio over here. Okay, silent wild types across all the positions. Okay, in this case we used a 41-membered library. Okay, and what you see is very consistent tight binding. Okay, you have to trust me on that slide, but I'll prove it to you on the next slide. Okay, here's the entire library. Okay, so there are those wild types on that nice little plateau there. Okay, we found plenty of up mutations. Unfortunately for most people's libraries, an even larger number of down mutations, with most of them being kind of disastrously down. Okay, but those aren't overrepresented in our libraries because we're looking at them one at a time instead of in combination. Okay, so what we got out of this data set in basically a single experiment was we characterized this binding loop of Avastin. Here it sits right in the middle of the molecule. Okay, and like I said, we can generate a heat map that basically ranks every single mutation possible in this loop, okay, as to whether it's an up mutation, a neutral mutation, or one of the two kinds of down mutations, either not that bad or hideous. Okay, all the mutations were assayed in a single experiment. Okay, we got up, down, and neutrals. Okay, and what I'll show you next is the impact of the mutations consistent with other slightly slower platforms. Okay, and we discovered a lot more mutations than were discovered before. Okay, and so the first set of data, and probably the only set that I'll show you today in the interest of time, is we'll compare against Genentech's ALIScan methodology. Okay, that's a favorite in the industry. And essentially what they do is they go in and substitute an alanine for each and every residue across all 68 residues in the CDRs of Avastin. Okay, so each of these was constructed separately, each of them was created separately, and then each of them was assayed separately. And the data is expressed as the IC50 of the mutant over the IC50 of the wild type. So large numbers are bad binders, small numbers are good binders. Data from their publication is presented here. Okay, and what you can see is some mutations to alanine, this Y95 for instance, okay, is a disastrous mutation that destroys the binding of the molecule. Same thing at 100B, okay, when we change this serine to an alanine, okay, and same thing here. So we'll look at a few of these a little more closely, and we'll also look at one that really wasn't a bad mutation. In fact, this one is relatively neutral. And you see a few neutral mutations here, you see a few bad ones, and in black you see the guys that are in between. This is why combinatorial libraries are so bad, is because most of the mutations are bad. So when you start combining things, okay, the vast majority of things in your library become bad. Okay, so like I said, we'll look at a few of these. I'll stop after 3 because everyone will start to doze off. But what you have are basically lists of amino acid changes by position. Okay, there's the wild type amino acid. Here are all the substitutions we looked at, and here's what their binding efficiency is. Okay, and in this one, this was a disastrous alanine mutation, and we confirmed that. Okay, but one thing that you find is there is a good mutation here that will actually increase binding affinity and would be a nice substitution if we wanted to use that in creating a biosimilar or biobetter molecule. Okay, there's another one that's a nice neutral mutation, but pretty much everything else is bad. Okay, here's a change at 100D where looking at the trip, okay, it's really the only guy that fits in there very well. I mean, things much below 0.9 are bad mutations, okay, and things below 0.5 are disastrous mutations. So we confirmed that their alanine mutation was a bad mutation, okay, or a disastrous mutation, and really nothing else will fit in that position. Okay, that contrasts with glycine at position 100 in this CDR. Okay, and what you can see are there are plenty of substitutions to make here. And in a minute I'll show you the structure of the molecule and you'll see why that is. Okay, but basically there are plenty of up mutations we could make here. There are plenty of neutral mutations we could make here. Okay, and we confirmed again that their alanine mutation was relatively neutral. In fact, exactly neutral in our data. Okay, here's kind of a summation of the data from one experiment, and that is, you know, surprisingly to what most people will tell you, we can make changes in CDRs of antibodies. Okay, they're not wildly common, but they're common enough that we can use them for a lot of engineering goals. Okay, and so if our goal here was to increase affinity, I've already shown you some mutations that can increase affinity. Okay, some of the ones that we found are the ones that are now used in Lucentis, okay, which was the affinity matured version of Avastin that they use as a monomer in the eye. Okay, we found a lot more up mutations, so we could generate a higher affinity version of that if we wanted. Okay, but a number of these are neutral mutations, and if you want to deimmunize a molecule without changing its bioactivity, those are the mutations you want to look at. Okay, and those are really naturally hard to select out of standard phage libraries, because you can really only select for ups in phage libraries. In these libraries, we're basically running a screening system, so we found ups, downs, and neutrals. Okay, as I promised, here's a kind of look at the structure. Okay, in sticks is VEGF, in balls is Avastin, and what you see is down at the core of the binding site, these are very, very unmutable positions. Basically, we can't change them at all, or we lose binding efficiency of the antibody. Okay, around the periphery, we see some that are a bit more plastic, and we can mutate more and still retain binding affinity of the antibody. And out at the sides here, we can see ones that really aren't interacting with VEGF at all, and our data shows that those are highly plastic and we can substitute in lots of changes there and not destroy the binding of the antibody. Okay, so here's a quick Avastin summary. If we have time at the end, I'll show you our comparison to Lucentis. But basically, we looked at 500 mutations at once. Okay, we found ups, downs, and neutrals. Okay, the silent mutations, essentially the silent wild types, basically all look the same in this system. So that's basically what you'd expect, and that's what our key validation QC is. We can also look at similar amino acid changes because the code is redundant, and so we can look at similar changes coded for by different codons and show what their covariance is, and the covariance is very tight. Okay, I showed you the quantitation of the mutations is consistent with the co-crystal structure. It's also consistent with the ALIScan data that Genentech provided. Okay, and we found a lot more information, and in some positions you would have guessed from the Genentech data. So we've got 15 additional ups that they didn't find, okay, and 19 neutrals that basically couldn't have been found with their phage display system. Okay, our lawyer likes us to put this in. This is, one of the key features of the system is now we can really protect a much wider array of biobetter molecules around our clinical candidate. Okay, and so instead of just patenting the sequences and the CDRs, we can patent all of the variants that will produce either a similar molecule or a better binding molecule. Okay, if we have other data on immunogenicity, okay, we can include that data for even stronger patent position. Okay, we can also use these libraries against a wide variety of targets and look at cross-reactivity. Okay, and this is especially useful in early preclinical candidates to helping change molecules that don't cross-react with your preclinical candidate to molecules that do cross-react with your preclinical candidate. Okay, we can look at stability, and we can look at, for the interest of Pfizer, pH sensitivity. Okay, so how would you apply this to immunogenicity? Okay, this has already been all talked about before, but immunogenicity of even so-called fully human antibodies, and this was my question earlier, are any of the therapeutics on the market actually encoded in the genome? I believe the answer is no. They're all affinity matured, so they're all basically looking like mutant proteins or foreign bodies when you look at a human. Okay, so immunogenicity is an important problem even with so-called fully human antibodies. They were derived from human genes, but they are not human genes. They're mutants. Okay, and as you know, these are our concerns. Okay, as you very nicely explained, Jeremy, all of these seem to be very strongly controlled by T-cell responses, and we'll talk about this in a minute. And reducing immunogenicity is part one, finding those immunogenic epitopes, which are linears, and then modifying the epitopes with the data that I just showed you to produce a molecule that binds similarly but has reduced immunogenicity. And before I go on, most of this data is going to be coming from Fiona's lab. Okay, so here's one of the first slides. It's very similar to the slide you just showed. Essentially, we start with a protein. In this case, it's an antibody. As I mentioned, antibodies therapeutic antibodies tend to have mutations in their CDRs from germline. Okay, antigen-presenting cells process those into peptides. Okay, here is kind of the key step in the whole process. Okay, if that peptide can be presented by MHC to a T cell that can respond, okay, that drives this whole downstream reaction and leads to the unfortunate consequence of anti-drug antibodies, and that's what we really don't like. Okay, so here's a set of data from Fiona's publication that basically showed if we look at 8 different humanized antibodies, where do we find the immunogenic epitopes? Okay, the way she presents the data is a little bit different than the way you guys present it. This is a T-cell assay, and essentially what we're looking at here across the x-axis is the degree of stimulation of the T-cell population from the whole experiment in average. Okay, and what we're looking at here is the number of donors, naive donors in this assay that responded. Okay, and so what you don't like, each of these little boxes is a peptide. Okay, the pink ones are peptides that contain CDR sequences. The blue ones are the ones that are framework only, And what you'd expect if humanization is working, and it does, is that the CDR-containing peptides are the immunogenic ones, and the framework peptides are right down here where you want them to be, closer to the origin. Okay, so our strategy for basically fixing or reducing the immunogenic risk of an antibody is to first, we let Fiona identify the T-cell epitopes. Okay, what we did is we ran similar assays to the mapping system that I showed you. Okay, like I said, that'll be published in a month or two. It's Abbott's secret now, and you'll have to wait till the publication comes out. Okay, we would identify neutral mutations from that data set, okay, in the regions of her immunogenic epitopes. Okay, that allowed us to essentially generate variant peptides. She'd rerun her T-cell assays, tell us which of those peptides had lower immunogenicity. We'd create variant proteins out of that, and then we'd test those proteins in functional assays. Okay, it seems like this is the only biotherapeutic that's out there, and that's Humira. Everyone seems to work on it. Okay, this is one of the reasons it's of such interest in this community. And I think this is actually what's written on the label. Correct me if I'm wrong. But the problem with it is if you administer it nice and consistently and keep the drug concentration in the blood up, okay, you have reasonable immunogenic responses. Okay, if you let patients take it home and the blood concentration is allowed to vary wildly, okay, immunogenicity, that's exactly what a nice parasite would look like, and immunogenicity goes up. Okay, and in this case, there's been a nice set of data showing that the development of neutralizing antibodies correlates with lack of drug response in rheumatoid arthritis. So this is a worthwhile problem fixing. Okay, here's Fiona's data, and we actually found immunogenic epitopes in a slightly different position than you. Okay, we found them in the light chain, and specifically CDR1 of the light chain. These two peptides were almost right next to each other. Fiona tiles her peptides very similar to the way you do. 15 mers offset by 3. And I think these guys were, for instance, 1, 2 was out, and 3 was there. So they were kind of the two ends of the 15 mer. Okay, so these are the bad guys. These are the guys we're going to target. Okay, Fiona presented this data way back in 2009. Okay, and this is not Humira's heat map. I don't have access to Humira's heat map anymore, so this is just a standard heat map, but this is where we would tend to plot the light chain CDR1. Okay, yeah, T-cell epitopes are linear epitopes, so we just line them up with the heat map. Okay, we read across the heat map to find functional neutral mutations, which in this case are basically light yellow or green. Okay, and so we can see a number of them here that would be good viable substitutions. If you wanted to increase the affinity of this molecule, you could pick the blue substitutions. Okay, and basically the same day she had the epitopes, we could design the peptides with this map. Okay, so she designed 77 variant peptides, both of single amino acid variants and double amino acid variants. Okay, she replanted them in her T-cell proliferation assay, and here's that data. Again, it's the same type of chart, so this is the degree of stimulation of the T-cells. This was the number of patients responded. Okay, the thing that was interesting to me is here are the same two wild-type peptides again, and they both come out very similarly to the first assay as being immunoreactive, is very few of the changes made things worse. Okay, most changes actually made things better. Okay, and so these are relatively straightforward to engineer if you just have an assay to tell you which mutations are going to be neutral here. Okay, and so the key thing from the engineering system was, what do these guys look like? Basically, the antibody engineers said all of these would be relatively similar in binding affinity. Okay, so we'd have a relatively functional antibody out the backside. We rebuilt antibodies out of all 10 of these peptides. Okay, and we stuck them with the wild-type heavy chain. Okay, the antibodies were purified and used in the TNF inhibition assay. And here's the data. Okay, there's the parent. Okay, naturally it works fine. We normalized everything to his response. okay, or her. And these variants in the middle, okay, turned out to be very nice variants, very similar TNF inhibition functions. All of these really would have passed our PDL humanization assays. All of these retain binding fairly nicely, but these four naturally are the best, okay, and they become candidates for the deimmunized version of Humira that Abbott now retains. Okay, so the key parts of the system is it's really a two-part system. Fiona's immune data told us where were the immunogenic epitopes in the molecule. Okay, it also told us once we designed variants, which were the lower immunogenicity variant peptides. Okay, the heat map told us which peptides we could make that would not destroy the binding site of the antibody. And then together, these two sets of assays pointed to 4 Humira variants that had wild-type binding activity and reduced immunogenic potential. Okay, so quick summary. Okay, this is a very rapid system since it's phage-based. It's very inexpensive as well. Okay, and so we can essentially map an antibody in about 3 months, okay, and gain basically a good feel for binding of every variant that you can create in that binding site. Okay, we can look at a variety of targets. Okay, we can combine this with other high-throughput assays to meet a variety of engineering goals, and those goals are listed here. Affinity optimization is naturally a standalone. If all you're interested in is binding affinity, this system can work all by itself. If you're interested in deimmunization, we need input from an assay like Fiona's or Proimmune's. Okay. If you're interested in cross-reactivity, I think this is an interesting application. Really all you need is the one library that we make, and we run it against 2 different proteins, 3 different proteins, 4 different proteins. We can overlay the map and basically tell you what each mutation does to each of those protein bindings. Okay, and like I said, actually it's not just Pfizer, it's Chugai. Okay, it was the other one that very nicely engineered proteins with respect to the pH of their binding. And so if you want to run these assays at different pHs, the same library, you can go ahead and do that. What they were interested in was antibodies that bound nicely at neutral pH, but then release their target in the endosomes. So at low pH, you can find those types of changes here as well. Okay, so there's the talk, but let me just do my acknowledgements. Facet was basically a later version of PDL, so basically the same company. This was Fiona's lab there, and Fiona ran her group. These 2 young ladies basically did immunogenicity assays. She ran the PXP system, which was our earlier version of the mapping system at PDL, okay, and Dixie ran the sorter, which was required for the system. Veronica did the data analysis both at Facet, and she's now at Full Spectrum Genetics running these assays with us. And that's it. Questions?